Hello students, welcome to Edupedia Word Videos. My topic for the presentation is Anatomy of Flowering Plants. In this section of the video, we will be discussing about the anatomy of dicot root. This is the sixth section of the chapter Anatomy of Flowering Plants. Flowering plants, they are also called as angiosperms students. Okay, so in this section of the video we will discuss about the anatomy, that is the study of internal form or feature of dicot root. Okay students, for better understanding of tissue organization of root, it is convenient to study the transverse section of the mature zones of root. Okay, so we get cells after cutting transversely, and this we observe under microscope. Okay, so let us study the anatomical feature of dicot root from out to in. Okay, so this is the outermost layer is epidermis. Okay, and it is also called as epiblemma. It is also called as piliferous layer, and these are the root hairs that are present on the epidermis. And root hair function is to absorb water and mineral, and these cells, they are called as cortex. The cortex, it consists of several layers of thin-walled parenchyma cells with intercellular spaces, and the innermost layer of the cortex is called as endodermis. Okay, but this is exodermis because it is present outside. And cortical cells, they store food such as starch, and this is endodermis, which is present inside the cortex, and it is comprised of single layer of barrel shaped cells. This way you can find it that it is dicot root without any intercellular space. And the tangential as well as radial wall of the endodermal cells, they have deposition of water impermeable waxy material suberin in the form of Casparian strip. So these are the barrel shaped cells, and the radial walls, they are made up of Casparian strip which are composed of suberin, and they are called as facet cells. So epiblemma, it is the outermost covering of the root, which is formed by a single layer of compactly arranged barrel shaped cells, and they are parenchymatous in nature, and the cells, they are characteristically thin-walled since they are involved in absorption of water. And some of the epiblemma cells, they are produced into long unicellular projections, which we call it as root hairs. That's why the outermost layer is called as piliferous layer also. Okay, because of the presence of the root hairs. So another is cortex. It is the major component of the ground tissue of the root, and it is represented by several layers of loosely arranged parenchyma cells. So this has parenchyma cells, and intercellular spaces are prominent in it. As you can see that these are the intercellular spaces which are present between the cells of the cortex. Okay, so the cortex is mainly meant for the storage of water, and the cells, they also allow a free movement of water into xylem vessel. Okay, now comes endodermis, that is another layer which is present inside the cortex. So they are compactly arranged barrel shaped cells. By barrel shaped cells, you can make out that it's a dicot root. Okay, because barrel shaped cells are formed in the dicot root. Some of the cells in the endodermis, they are thin walled, and they are known as facet cell. I have just shown you in the diagram. And facet cells, they allow water to pass into the xylem vessels, and the remaining cells in the endodermis, they are characterized by the presence of thickening on their radial wall, and these thickenings, they are known as Casparian thickenings, and they are formed by deposition of waxy substance which we call it as suberin. I have just shown you in the diagram. Now steel is what? It is made up of three things such as Peri Cycle, conjunctive tissue, and vascular bundle. And Peri Cycle is the region that lies immediately below the endodermis, and it is represented by a single layer of parenchyma cells. Now vascular bundles, they are described as radial and tetrarch. There are 4 bundles each of xylem and phloem that occurs alternatively, and they are described as exact position. I have already taught you endark and exact position of xylem in my previous section of the presentation, so for better understanding you can refer to it. Now let's talk about the conjunctive tissue, which is present in dicot stem. It is represented by a group of radially arranged parenchyma cells which are found in between vascular bundle. I'll show you what it is. So this is unicellular root hair which is present on the epiblema. These are the starch grain that stores the starch. This is the metaxylem, which is a smaller dot, and this larger dot is called as protoxylem, which is formed in the early ages, and metaxylem it is found in the later stages, and this is the Casparian strip. Okay, and this is conjunctive parenchyma, this is phloem. Okay, and this is sclerenchyma, and this portion is called as Peri Cycle, and this is what endodermis, this layer, the which is represented by this lighter color. So let's talk about the diagnostic features of dicot root. Presence of thin walled cells in the epiblema, so these are called as unicellular root hairs. Absence of cuticle and stomata, presence of unicellular root hairs, absence of hypodermis. See, no hypodermis is there. Can you see the hypodermis here? No. Presence of passage cell and Casparian thickening in the endodermis. It is a unique characteristic of dicot root. Presence of uniseriate pericycle, which is made up of parenchyma. As you can see that this is the Peri Cycle, which is uniseriate, and absence of pith. Can you see the pith here? No. So no pith is there, and the presence of radial vascular bundle that exhibits tetrarch condition with exact xylem. Okay, so this is the single layer. So this is the single layer of parenchymatous cell, which is Peri Cycle. This red color is representing Peri Cycle. It surrounds the stele. Stele is composed of 3 parts: metaxylem, and this is protoxylem. Okay, so protoxylem it is present outside and metaxylem is present inside, hence this condition is called as exact xylem. Okay, so recollect that roots show radial vascular bundles. So this is like this. Okay, so this is radial vascular bundle, I have already taught you. This is phloem, and this is xylem, and they are present in the same radii. That's why it is called as radial vascular bundle. And the rest of the cells, they are around vascular bundle, they are parenchymatous in nature. So this is parenchymatous cells which are present outside the xylem and the phloem. So this is also called as connective tissue, and it is also called as conjunctive tissue because it is found between xylem and phloem. So connective tissue is also called as conjunctive tissue, which is found between xylem and phloem. Okay, and this is the central part of the stele, if present, it is very small. Okay, now we'll see the transverse section of the dicot. This is the epiblema with root here, and this is the exodermis, which is represented by this red color, and this yellow color is representing cortex, and this is endodermis, which is made up of barrel shaped cells which are suberized, that because of the deposition of suberin. And this is the Peri Cycle, which is represented by this red color, and this is protoxylem, this is metaxylem, this is phloem, and it is radial vascular bundle, it is connective tissue, and this is the pith. So number of the group of xylem and phloem is equals to stele. It may be diarch to exact. Okay, 1, 2, 3, and 4. Okay, so stele here in this case is tetrarch. Now we'll talk about the anatomy of root, students. So for better understanding of tissue organization of a root, it is convenient to study the transverse section of the mature zone of these root. Okay, so this is it. Let us study the anatomical feature of monocot root from out to in. See, this is the epiblema, and it has root hairs. That's why it is called as piliferous layer, and it helps in the absorption of water and mineral ions. And this is what it is, cortex, and this is exodermis. So cortical cells, that is cortex, they store food material, to be more precise starch and water. And this is endodermis, which is made up of barrel shaped cells And radial walls, they are made up of Casparian strip, which is composed of suberin, and that is called as passage cells. Okay, so epiblemma, in case of monocot root, it is outermost covering of the root, which is formed by single layer of compactly arranged barrel-shaped parenchyma cells, and they are very thin wall since they are involved in absorption of water. Okay, and cuticle and stomata are absent in the root. Some of the epiblemma cells, they are produced in long unicellular projection, which we call it as root hairs. Hence, epiblemma is known as piliferous layer. Now we'll talk about the cortex, which is this major portion. It is also called as bulk tissue. It is represented by several layers of loosely arranged parenchyma cells. Intercellular spaces are prominent, and cortex is mainly meant for storage of water. And the cells, they also allow free movement of water into xylem vessels. Now this is endodermis. It is the innermost layer of the cortex, which is formed by compactly arranged barrel-shaped cells, and some of the cells in the endodermis, they are thin-walled and they are called as passage cells. Passage cells I have shown you in the diagram. It allows water to pass into the xylem vessel, and the remaining cells in the endodermis, they are characterized by the presence of thickening on their radial wall. And these thickenings, they are called as Casparian thickenings. I have already shown you in the diagram. Now we'll talk about the stele and Peri Cycle and conjunctive tissue. Stele is the central cylinder which consists of Peri Cycle, conjunctive tissue, pith, and vascular bundle. Now what is Peri Cycle? It is the outermost covering of the stele, which is represented by a single layer of parenchyma cell. And conjunctive tissue, it is represented by loosely arranged parenchyma cells, which is found in between vascular bundle, and it is specialized for storage of water. See, this is the Peri Cycle, which is represented by this red color cells. Okay, and this is the outermost layer, as you can see, and it is made up of single layer of parenchymatous cell. So this is Peri Cycle, which surrounds the stele, and the stele is composed of metaxylem and protoxylem. See, smaller dots are called as protoxylem and bigger dots are called as metaxylem. So here in this, xylem is exact position because root shows radial vascular bundle, so they have phloem in the same radii, okay, as with the xylem. So the rest of the cells around the vascular bundle, they are parenchymatous like this, and it is also called as connective tissue. It is also called as conjunctive tissue, which is found between xylem and phloem, and it has pith. So central part of the stele is large and well developed. So this is the transverse section of monocot root. This is epiblemma with root hair, then exodermis, this is cortex, and this is the endodermis, which is suberized because of the presence of suberin, and this is the passage cell, and this is the Peri Cycle, protoxylem, metaxylem, and phloem, connective tissue. So this is the transverse section of monocot root, and please note that pith is very larger in case of monocot, and number of the group of xylem and phloem, as you can see, it is more than 6. 1 2 3 4 5 6 7 8. 8 are there. That's why it is called as polyarch. 1 2 3 4 5 6 7 8, and the stele is polyarch. Now anatomical differences between two types of roots, let's see. So this is the transverse section of dicot root, and this is the transverse section of monocot root. So 1 2 3 and 4, it is tetrarch, or you can say it is between diarch to hexarch, and this is polyarch because it is more than 6. Okay, now connective tissue is parenchyma, and here in this case it could be parenchyma also, it could be sclerenchyma also. Vascular cambium is present in dicot root, but vascular cambium, it is not present in monocot. Pith, as you can see, it has larger pith. Diagnostic feature of monocot stem: absence of trichome, there are no trichomes present, presence of stomata, presence of hypodermis, which is made up of sclerenchyma, presence of undifferentiated ground tissue, as I have shown you in the diagram, and numerous vascular bundles, they are irregularly scattered with the centrifugal arrangement, and vascular bundles are conjoined, collateral, and closed type. Okay, presence of only 2 protoxylem and 2 metaxylem vessel in each bundle is seen. Presence of lysigenous cavity is also seen, but no phloem parenchyma is seen. Okay, so these are the diagnostic features of now this was all about it. In my next section of the presentation, we'll be talking about the monocot and dicot stem. So till then, stay tuned and keep watching Etopedia word videos. Thank you.